Here is something which I find fascinating. To be able to watch a living retinal cell receive a photon of light. Isn't that extraordinary? I mean, in the past, when you and I qualified as medical students, we had to freeze these specimens and look at them under an electron microscope, and very sterile they were. But here we are, able now to observe things that are happening at wavelengths smaller than the wavelength of light. This is fascinating physics. We can see individual receptors in the eye ignite as a photon hits them. That means that we can begin to put drugs into a living retina and watch the effects of those drugs on living cells. We can observe the process inside each cell. This is fascinating, dramatic, life-changing, revolutionary technology which opens up a vast new area for research. In the past, you've had to do all kinds of experiments, then you kill the animal and you go and look. Now we can do it in real time on tissue we can observe. Fascinating. And then, of course, there's visualization, the kind of things, again, which we heard about just now. To be able to travel inside different parts of the eye in 3D. Amazing. To be able to walk up a blood vessel inside the heart and look at the atheroma in 3D and in color. Fascinating. To be able to train not just a couple of people in endoscopy using microscopic instruments, but maybe 1,000 people at the same time to do extraordinarily sophisticated operations within the eye. Using 3D robotics, 3D imaging, robotic-assisted and enhanced surgery. Amazing stuff. To be able to model things in 3D. To be able to take an extraordinary new revolutionary lens and rotate it around within the eye of the patient just to see how the pieces will fit together. To be able to use augmented reality, to impose a computer-generated image of what might be, or a piece or a device you're going to insert, and to create exactly the right space for it with microscopic devices. Extraordinary, fascinating stuff, exciting stuff, and we've only just started. And, of course, this is why we're seeing so many advances in terms of the ingenious surgery that's happening inside the eye. The ability to find all kinds of new solutions for glaucoma, to decompress an eye. To find all kinds of amazing new ways to resculpture, to reform the substance of the cornea itself. We've already heard about all kinds of lenses which at the moment these are prototypes. They're relatively primitive compared to what we will have in 20 years' time. But to be able to put a lens in which allows us to focus simultaneously at different distances is something that was beyond comprehension 20 years ago. I'm asking you the future. I'm only an expert outside of your industry. But you futurists, where do you think that lens will be in 25 years' time? My goodness. We'll be in a fascinating place. Now, there's a great debate about whether we will ever be able to form a lens that is as beautifully made as the human lens, that would be able to be attached as well as a human lens to the iris and to be able to adjust properly. You know what? I think it will require a miracle of science to do it, but you know what? I'm sure we'll manage it. I look at the speed of innovation that I'm seeing in every other part of science with nanotech, infotech, biotech, and just about every kind of tech you can imagine. It can't be beyond the wit of human beings one day to be able to create a capsule filled with some kind of transparent jelly, which is as beautiful and as gentle and as pliable and has exactly the same kind of characteristics as a perfect young human lens. It may take us 2 decades, but I'm sure we will get there. And I'm also sure when we've made it, it will cost a fortune to make the first 20, and a smaller fortune to make the next 100, and almost nothing to make the following 1,000,000. Because we're getting good at these kinds of things. Here is an extraordinary piece of science. I was talking to one of the professors in Manchester who's installing these things. Most of you will recognize this, of course. To me, it was new. What is it? It's a telescope. What it does is it focuses light that comes in through the iris and diverts it so it lands on a piece of undamaged retina. It's designed for someone with macular degeneration. Fascinating. Enables people to see by bending light through the eye. Ingenious. And then there are smart devices. You know, this is a huge market. You've heard about wearables. We're looking about a market of 250 million wearable digital devices by 2023 being sold every single year. This is a market that will be worth about $70 billion a year by 2025. And a number of these devices, a significant number, will land up inside or close to the eye. Let's have a look. One of them, of course, is Google's great piece of engineering. This is fascinating. This is history. You tell me the future. This is today. You tell me the next 30 years. This is just the same technology as in your credit card. Did you know that? In your credit card, you have a grain of sand. It's a chip the size of a single grain of sand. It has a microprocessor, a memory. It has an aerial. The credit card has an aerial. When you go with a payless payment in the bank, that's what you do. This aerial here collects a power surge, electromagnetic power, from an external device. This powers the chip up. It wakes up and says, hello, I'm here. It says, send me your data. It says, what data? Has a look, collects some data, and sends it out. In this particular instance, it will be sampling human tears for glucose, sugar. So it will be continuously sampling. As often as your iPhone wants a sample, it will continuously sample your blood glucose level. I think that's cool. And to do it through something that is completely non-invasive, and yet is taking a minute sample from something which is representative of what is in your blood, is totally ingenious to me. And I think of other things I would love to sample at the same time. Can you imagine being able to get a printout instantaneously of a whole range of different substances that just happen to be present in tears in a way that correlates to the level in blood? Fantastic science. Listen, I'm not telling the future. This is history. You tell me the next 20 years. We can go further. What about this one? This one does look a little science fiction, but it's already here. This is history. This device has been made by the Pentagon. It's a transparent polymer. On it, it has nanoscale phantom dot light-emitting diodes. What they do is project microscopic dots of light direct to the back of the retina so that you get an image. What do you get an image of? It's designed for fighter pilots, so they get a head-up display, not through some kind of screen or anything like that. They get a head-up display wired into the back of their eye from the lens itself. Again, using the same kind of wireless thing. Fascinating. We're just on the edge of extraordinary science. And then what about detecting pressure in glaucoma? Wouldn't it be nice for the iPhone to be able to tell you every hour what is actually happening to the pressure inside the eye? My friends, that's history too. Devices have already been made, they're in prototype, which will sit inside the eye and be powered by sugar. Interesting stuff. Actually, we can go further. Now we're into really old tech. The idea of creating another chip and putting it either on the outside or the inside of the retina, where it connects up. And what we're doing here is we're allowing the chip to become the photoreceptor. The chip produces a current as a response to that, which then is detected by the wiring on the retina itself. These things have been around for a very long time in different experimental forms. The only challenge is the acuity. How many little electric sensors can we pack into 1 square centimeter? And you know the answer to that. It will be more tomorrow than today. And what kind of acuity will we get? I'm not quite sure. Could we increase the acuity? Yes, we could. At the moment, as you know, almost all of our ability to see is focused on the microscopic area of the retina. I can imagine the lens being used to actually scatter an image perhaps over quite a large chip, a much larger chip than you would usually use, and get all kinds of other data. It's crude. Would I want one in my eye? Yes, I might if I'd gone completely blind. Is it the best solution? No. This is already looking a little last century. You see, you have competition, a wave of technology that sounds smart, a little science fiction, but it's already out of date almost by the time it reaches mass production because there's something even better coming up behind it. And what is that? Probably stem cells, which have been in the news so much in the last 2 days, I really thought we should talk about it some more. So for a long time now, I wrote a book in 1993 called The Genetic Revolution. And I predicted in that we would be building all kinds of things using stem cells, and indeed that has turned out to be the case. In 1998, I wrote another book called Futurist, which described the use of stem cells in all kinds of situations. And we are now routinely in London and in Birmingham using stem cells to help repair. I say not repair. We're not sure even how they work. You can inject stem cells into damaged heart, and the heart beats better. That's a fact. What we're not quite sure about is whether the stem cells actually transform into heart tissue or whether they just produce cytokines and then the heart recovers. Who cares? I couldn't care less. There's this great debate. Some say stem cells are nonsense, they don't work. Others say stem cells are regenerative. I couldn't care less. All I want to know is, does it work for the patient? And the answer is absolutely yes. So let's not get into a technical debate about whether when you inject stem cells into a damaged eye, they migrate and produce whatever they're supposed to produce. Let's just see what happens as a result, and the results are fascinating. We are seeing sight in completely blind animals as a direct result of stem cell experiments. Over 10 years ago, as part of research for a pharma company that I was with, because I lecture very large corporations on global trends all over the world in every industry. Over 10 years ago, in 2003, I was first in contact with a researcher at Harvard Medical School who was already restoring sight in mice and rats, routinely using stem cells. Hello? That's kind of exciting, is it not? He just needed ethical permission before he dared to do it in a human being. Because stem cells have a habit of going out of control. If they go out of control, they grow and they don't stop, and you die of cancer. So you take care. And what's happened has been slow. But before we come back to that, let's look at some other things. I've got a 3D printer at home, but printing toys is really boring. I want to print pieces for people. Can we? Yes, we can. Here is a piece for a person. It's been printed in a bubble jet printer. Instead of the colors, you just fill cartridges full of jelly and certain cells, whether it's cells to produce cartilage, cells to produce red blood vessels, whatever it is. And you just spray, zoom, zoom. Lexmark produces bubble jet printers you probably have one in your home. They've produced a very big one for printing skin. You take a burns patient, put the leg under, zoom, 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 zoom. It just sprays layer after layer after layer of perfect skin. Isn't that fantastic? You're allowed to get excited? Yeah. Come on. Yeah. This is cool stuff. I tell you, if you have a child who's been severely burnt, you would be very grateful for Lexmark 3D print technology, would you know? If you had a child who had a terrible cancer, which meant that he or she would never, ever be able to breathe on their own without some kind of metal prosthesis, which would only last a little while, and you have the capacity, using the child's own cells, to grow a new piece on a cartilage scaffold or something like that. This, I have to say, is fantastic news. Can we do the same in the eye? Let's not hype it too much, but let me tell you history, then you tell me the future. Here is history. Here is a chip. On the chip is all kinds of exposed pieces of the chip surface. It's a microchip. Here are brain cells growing onto the surface of the chip, stem cells growing onto the surface of the chip, starting to communicate, putting little branches out and forming bandwidth. They are talking to each other. They are not only talking to each other, they are talking to the chip. Each time they grow along the surface of the chip, they detect electrical current. They think, I found another nerve. And they put even more bandwidth onto that connection. This, my friends, is a biodigital brain, formed using human tissue. We can implant chips inside people's heads, and your brain knows precisely what to do with them. Your brain is genetically programmed to communicate with chips, it turns out. We have mice and rats sending messages virtually to each other across the world. We have connected 4 brains together of different rats, where one rat had to see something, one rat had to hear something, one rat had to move a lever. They had to learn to cooperate in a neural network. Do you know what? They started to solve problems together through wireless communication. Put your hands up if you find that a little scary. Put your hands up if you would like to have a biodigitally enhanced brain. 1 or 2. Put your hands up if you would be very unhappy for your children to have a biodigitally enhanced brain. See, we're not quite sure about this kind of stuff. I tell you, we're on the edge of extraordinary science. Now, if you do this, it means we have the capacity to put one of these chips in the optic cortex. It means we can deliver whatever visual data you like right where it's needed. Would it be a sufficient resolution? Not so sure. But now you can begin to see the idea of putting a thing in the back of the eye. Well, there's another solution, but they're still pretty crude. I'm actually much more interested in growing parts of the retina. The first way to do it is tacky, which is to take pieces of embryo that have been aborted, which is illegal in some countries, and to many of your patients is distasteful at best or makes them feel extraordinarily uneasy at worst. They do not want to have pieces of aborted fetuses inside their body. And the good news is they don't have to. Again, I predicted in 1993, again in 1998, and it's been proved many times since, that more or less whatever you would like to do with an embryonic cell, we can do with your own tissue. And how do we know that? I can prove it to you with Dolly the sheep. Do you remember Dolly the sheep, the cloned animal? Well, Dolly the sheep, to remind you, you take a skin cell, you remove the nucleus from the skin cell, which contains all the genes, of course, except the mitochondrial genes. You then take an egg, remove the nucleus from the egg and throw it away. You put the egg from the skin cell, you put the nucleus from the skin cell inside the egg, put a spark of electricity. That egg, the chemicals in the egg are now bathing the nucleus, the genes, from a skin cell. The chemicals in the egg tell the skin cell genes that it's not a skin cell, it's an embryo. And so, out of that skin cell genetic code, we start to get an entire cloned animal. Pop it in the womb, and it gets born as the identical twin of the person from whom the cell is taken. And we've done that quite routinely in humans, but not implanting the embryos. So it's obvious that I can get whatever cells I want out of your body. All I have to do is take any cell from your body, skin will do, and make sure that the chemicals in that cellular cytoplasm are the right ones to stimulate the genes in that cell and persuade it it's a more primitive cell. It's just a question of the right chemical bath. And I don't want to create a whole embryo, which is a bit tacky. I don't want to create a clone of you. I don't want to create your twin brother by mistake. All I want to do is find a way to create some cells that might think that they could be persuaded to become retina with the right chemical bath. Because Dolly was already old genetically on the day she was born, because one of the things we hadn't quite sorted out was the aging of her genes. The genes, when she was born, were as old as her, and you know the thing about telomeres, when cells divide, the genes shorten and things like that. So she had premature aging problems, which is something we can overcome. But this is what we have done in humans, not using embryonic stem cells, and you know this story. This is red photoreceptors rod photoreceptors in green within a mini retina formed in the laboratory from induced pluripotent stem cells. That is, they're adult stem cells that have been bathed in juices to try and persuade them that they're more primitive than they actually are. And they've behaved beautifully. And we've seen retinal cells created from adult skin cells through this kind of process. You then form a sheet of these retinal cells. You roll up the sheet, you make a tiny little nick. You know this story. You make a tiny nick in the eye. You slip the sheet into the eye, and you try to persuade the sheet to stick. We've also done other experiments where people have just injected stem cells fairly randomly and hopefully near the right place, and they found another interesting thing. Where the eye is damaged, the stem cells migrate, and they start to incorporate themselves and look like they're developing beautifully into the right new retinal cells, and they're wiring themselves up to the optic nerve. But we've seen very poor results from that in terms of vision. So that's why people are now growing whole sheets of it. This stuff is working. We've had people who have had, actually, this is not new. I mean, you know we can do this in you anyway. We can cut out a piece of retina. I know it's easy to say and tough to do, but we can cut out a piece of your retina for one part of the eye, and we can paste it over another part of the eye. If it sticks and it works, you get sight back for years sometimes. So we are right on the edge of extraordinary science, which is being propelled by the urgent need to restore sight. It runs in parallel with stem cell advances elsewhere, and I think it's utterly fascinating. Now, of course, if you've got, say, retinitis pigmentosa, and you've got a condition which knocks off retinal cells in your genes, then you can create all kinds of new retinal material, and it may not last very long. We have the same problem with diabetes. In parallel to this, we are replacing insulin-secreting cells for people with diabetes. It's becoming routine. The only problem is, in juvenile diabetes, the immune system comes along and knocks off those cells. So you have to repeat the process. But you know what scientists have decided to do? They've found a way to create a sophisticated membrane that locks out antibodies and keeps the white cells away. Into the membrane will flow sugars, amino acids, and the other things to keep the transplanted cells alive. They're filling these bags stuffed full of cells from my own body which have been persuaded to make insulin again. Pop them in the bag, transplant them under my skin, and hey presto, I'm cured. Because it's an immunologically protected environment. Now, I don't know too much about the eye, but my understanding is that the eye is also a special environment. It doesn't have quite the same immunological processes attacking it, correct? So straight away, we don't perhaps need to put them in the same kind of bag. This science is beyond most of us to understand fully, and perhaps nobody on Earth understands fully how this will work. I'm just telling you history. You tell me the next 30 years, and I would suggest. I'm not saying that everyone like Julian is going to have restored sight. I wish that were true. But what I do believe is that we are going to see some extraordinary progress, not only in preventing different spheres of blindness, but in ways of managing them and enhancing. And I'm not saying people are going to get perfect sight back, but I do think we're going to find it relatively common that someone who's lost sight completely from a whole host of conditions, including, a whole cluster of them, will find that they have some acuity restored. And I think that is a truly extraordinary thing and much to be welcomed. Now, what does it all mean in summary? Well, to me, it means one big thing, and I've seen this elsewhere in other parts of medicine. Life is too complicated. You know, you can't do these things without a complete fusion of cutting-edge diagnostics, extraordinary microscopy, the very latest in medical research, fantastic surgical ability, extraordinary micro-instrumentation, the ability to take care of the whole person, to manage the other conditions that the person has, whether it's diabetes or glaucoma or any other things as well, to have every subspecialty present in the process of decision-making, and what is most important of all, without having the best-informed people right at the very first encounter when the patient presents and says, I'm just getting headaches, I'm not seeing as well as I was. And in that very first conversation, when you look in your microscope and you begin to make that diagnosis and you're thinking, okay, now how do I communicate this? And the very next two sentences you say will determine that person's journey, either perhaps a cycle of despair or a procession of hope. Either something that becomes a chaotic place of anxiety and fear, or a place of quiet confidence that somehow we will find a way through. And that's what I believe has made Optegra so special from what I've heard from you guys. It's precision, integrated, compassionate management. It's not just a hospital. That's only a tiny part of the whole picture. It's what you do in the GP surgery, what you do in Boots or in the local chemist or wherever it is. Just coming alongside an individual patient who's really worried that their worst fear might be about to happen. They're going to lose their sight. And to be able to take them at that point and that they know that they're now going to begin a journey with highly specialized people who will deliver absolutely world-class care at the very cutting edge of what is humanly possible. And I think that's a tremendous achievement.